In molecular biology, messenger ribonucleic acid is a single-stranded molecule of RNA that corresponds to the genetic sequence of a gene, and is read by a ribosome in the process of synthesizing a protein. mRNA is created during the process of transcription, where an enzyme converts the gene into primary transcript mRNA. This pre-mRNA usually still contains introns, regions that will not go on to code for the final amino acid sequence. These are removed in the process of RNA splicing, leaving only exons, regions that will encode the protein. This exon sequence constitutes mature mRNA. Mature mRNA is then read by the ribosome, and, utilizing amino acids carried by transfer RNA, the ribosome creates the protein. This process is known as translation. All of these processes form part of the central dogma of molecular biology, which describes the flow of genetic information in a biological system. As in DNA, genetic information in mRNA is contained in the sequence of nucleotides, which are arranged into codons consisting of 3 ribonucleotides each. Each codon codes for a specific amino acid, except the stop codons, which terminate protein synthesis. The translation of codons into amino acids requires 2 other types of RNA: transfer RNA, which recognizes the codon and provides the corresponding amino acid, and ribosomal RNA, the central component of the ribosome's protein manufacturing machinery. The idea of mRNA was first conceived by Sidney Brenner and Francis Crick on April 15, 1960 at King's College, Cambridge, while François Jacob was telling them about a recent experiment conducted by Arthur Pardee, himself, and Jacques Monod. With Crick's encouragement, Brenner and Jacob immediately set out to test this new hypothesis, and they reached out to Matthew Meselson at the California Institute of Technology. During the summer of 1960, Brenner, Jacob, and Meselson conducted an experiment in Meselson's laboratory at Caltech which established the existence of mRNA. That fall, Jacob and Monod coined the name messenger RNA and developed the first theoretical framework to explain its function. In February 1961, James Watson revealed that his research group was right behind them with a similar experiment in roughly the same direction. Brenner and the others agreed to Watson's request to delay publication of their research findings. As a result, the Brenner and Watson articles were published simultaneously in the same issue of Nature in May 1961, while that same month, Jacob and Monod published their theoretical framework for mRNA in the Journal of Molecular Biology.